Hey BIM 100 students, Professor Pasquinelli's final exam is right around the corner. So let's do a super fast run-through of the big ideas from lectures 13 to 17 to get you ready.
All right, let's dive right in. We're going to follow the story of our genetic information, starting with how it's controlled after it's been transcribed, then how we can measure all that activity, and finally, what happens when it all goes haywire, either from a virus or from within, like in cancer. So first up, regulating the genetic message. The cell doesn't just make an RNA and call it a day. It has this incredible toolkit of small RNAs to fine-tune the message after it's been made. And a key process here is RNA interference, or RNAi. You can think of it as the cell's search and destroy system. It uses these tiny double-stranded RNAs to find a messenger RNA that's a perfect match. And when it finds it, it's flagged for destruction. Simple as that.
Now this is a really important distinction for the exam. On one side, you have RNAi, which uses siRNAs. Think of it like a sniper. It needs a perfect 100% match, and its action is decisive. It slices and destroys the target. But on the other hand, you have microRNAs, which our own bodies make. They're more like dimmer switches. They bind imperfectly and just dial down the gene's expression.
And here's the assembly line that makes those microRNAs. It starts out in the nucleus as this long transcript, which gets a little haircut from an enzyme called DROSHA. Then it's shipped out to the cytoplasm, where another enzyme, dicer, gives it the final chop. And once it's ready, it gets loaded into this complex called RISC, and that's when it can finally go out and do its job. Okay, so we've seen how cells can control one gene at a time. Now, let's zoom way out and look at how we study the entire genetic library of an organism. Welcome to the world of genomics. And the main idea here is that genomics isn't just about one gene, it's about the whole shebang. It gives us the complete instruction manual for how to build and operate an organism.
And we have some amazing tools to do this. BLAST is basically like a Google search for genes. But then you have these super powerful techniques like ChIP-seq and DNA-seq. They let us ask two really important questions on a genome-wide scale. First, where are proteins binding to the DNA? And second, which parts of the DNA are open and accessible for business?
But just having the entire library of books, the genome, doesn't tell you what's actually important right now. For that, we need to know which books are being read. And that, my friends, is transcriptomics.
Here's the best way to think about it. Genomics is the library. It shows the cell's total potential, and it's pretty much the same in every single cell. Transcriptomics, on the other hand, is the list of books currently checked out and being read. It shows the cell's actual, real-time activity, and it changes constantly.
So how do we see what's being read? Well, a classic way is with microarrays. You take two samples, say one with a drug and one without, you label their RNAs with different colored dyes, green and red, and you wash them over this chip. The color tells you everything. If a spot is red, that gene was more active in your test sample. If it's green, more active in the control. And yellow? About the same.
But these days, the gold standard is RNA-seq. It's a total game changer. Instead of just seeing relative colors on a chip, RNA-seq literally sequences all the RNA in the cell. This gives us an incredibly precise picture of what's going on. We can even discover totally new genes or different versions of existing ones. It's so much more powerful.
Okay, so we've got the tools to see what a normal cell is up to. But what happens when that normal activity gets hijacked by an outsider, like a virus?
And wow, viruses are incredibly varied. Just look at this. Their entire genetic blueprint can be DNA or RNA, and that can be single-stranded or double-stranded. This diversity is their secret weapon. It gives them a whole bunch of different strategies to take over a cell.
But no matter what kind of genome a virus has, its mission always comes down to two simple things. Number one, make viral proteins. It's got to trick the cell's own machinery into building its parts. And number two, copy the viral genome. It needs to make more blueprints to stuff into all the new viruses. And for our last topic, let's look at what happens when the cell's own internal controls completely break down. That's when you get the uncontrolled cell growth we call cancer.
And here is probably the most powerful analogy to understand the genetics of cancer. You've got proto-oncogenes, which are like the gas pedal. They tell the cell to grow. And then you have tumor suppressors, which are the brakes. They tell the cell to stop. Cancer happens when the gas pedal gets stuck down or when the brakes get cut.
And this explains the genetics of it perfectly. For an oncogene, the gas pedal, you only need one copy of the gene to get a stuck-on mutation to cause a problem.
That's a dominant mutation. But for the breaks, the tumor suppressors, you've got a backup. You need to have a loss-of-function mutation in both copies to lose that protective break. That's recessive.
And a classic real-world example of this is the BCR-ABL fusion protein. This happens when two chromosomes literally swap pieces, creating this new Frankenstein protein that acts as a super-powered oncogene. It's constantly screaming, "Divide!" at the cell, and that's what drives certain types of leukemia.
So as you wrap up your studying, think about this. Everything we've talked about, from viruses to cancer, comes down to errors in these basic cellular systems. So if you had to pick just one process that acts as the ultimate guardian of the genome against all these failures, what would you choose? Okay, that's it for this primer. Good luck on the final.